In this video, I will be explaining the TRISOL protocol that we use to isolate DNA and RNA after brain dissections. So the TRISOL protocol we used on the team is adapted from Life Technologies' online TRISOL DNA isolation protocol. For this protocol, the following materials are necessary. A large supply of deionized or ultra-pure water, 95% ETOH, 100% ethanol, 1.5 milliliter tubes and homogenizers for those tubes, 8 millimolar solution of sodium hydroxide. And now to make 10 milliliters of this 8 millimolar solution, you can add 0.8 milliliters of 0.1 molar NaOH, which can be found in the chemistry department, and add 9.2 milliliters of deionized water to that. You also need PCR primers, which we obtained from Rutgers and Rockefeller universities. We called them and asked for zebrafish primers for PCR, and they were able to give them to us. Now, the primer concentrations that we used specifically for our PCR were 0.5 microliters of forward primer, 0.5 microliters of reverse primer, 1 microliter genomic DNA, 10 microliters of 2x master mix, and 8 microliters of ultra-pure water all mixed together. Now, before beginning, I recommend that you have an ice bucket ready to store all of these things, and that you gather all the materials that you'll need, like tips and pipettes, before you begin the protocol. It makes doing the actual experiment a lot easier. Now, step 1 is to add 1 milliliter of TRISOL to a 1.5 milliliter tube in the fume hood. I recommend you work in the fume hood for most of the lab because you don't want to be walking back and forth across the lab with TRISOL or chloroform open. Now to this 1 milliliter solution, you add the tissue sample and you homogenize it with the special homogenizers that we have for 1.5 milliliter tubes. Now this is a natural stopping point in the experiment where you can store the samples that you have in the -80 for at least a month. But if you decide to keep going, step 2 is to incubate the homogenized samples for 5 minutes at room temperature. So in other words, just let the samples sit for 5 minutes. Step 3 is to add 200 microliters of chloroform in the fume hood. This cannot be stressed more. You need to be working in the fume hood when you work with chloroform because any chloroform that you get either on your skin or that you smell or ingest into your body will knock you out. Now cap the tube securely and shake vigorously for 15 seconds by hand after adding the chloroform, and then you let it sit. You let it incubate for another 2 to 3 minutes. So meaning let it sit on the desk. Step 4 is to centrifuge at 12,000 RCF for 15 minutes at 4 degrees Celsius. The centrifuge that we used was the Eppendorf 5424R at the back bench. Keep the cap hinge on the outside, and the pellet should be on the same side as the cap hinge, so that if it's not visible, you'll know where the pellet should be. Now, after you centrifuge, the sample should separate into 3 phases: the red phase, which is the phenylchloroform phase, interphase, and a colorless upper aqueous phase. You should remove the colorless phase at a 45-degree angle and save the interphase and the red phase for DNA, and you want the RNA. This is also another natural stopping point where you can store at 4 degrees Celsius overnight. Now, make sure to remove everything above the interphase when you keep going. You want to make sure you get all of it so you don't contaminate your DNA. Step 6 is to add 300 microliters of 100% ethanol, and you want to cap the tube and invert by hand several times after adding. This is what inverting looks like. You want to just flip the tube back and forth. And then you let it incubate at room temperature for 2 to 3 minutes, so let it sit again. And then after that, you centrifuge the tube at 2,000 RCF for 5 minutes at 4 degrees Celsius, and a pellet should form. Now you want to remove the phenylethanol supernatant, the top layer, from the solution that forms after you centrifuge. We, for this experiment, skipped the DNA wash, but if time allows for it and if we have enough resources, I recommend doing the DNA wash before you continue so that you know you have a clean sample. But if you're going to skip the DNA wash like we did, the next step is to resuspend the DNA in 8 millimolar NaOH at 0.2 to 0.3 micrograms per microliter. So you want to use the sample that you had already made previously. You want to make it beforehand so that you don't have to try to make the NaOH sample during the experiment. And you want to add 300 to 600 microliters of this 8 millimolar NaOH. And now it's important to resuspend in a mild base, not water. That's why we're using NaOH. You don't want to use water for this. After that, you centrifuge at 12,000 RCF for 10 minutes at 4 degrees Celsius. And then you want to transfer the supernatant you get after centrifuging into a new that contains the DNA into a new tube. Now, this is also after this is a natural stopping point as well. DNA can be stored at up to in the 4 degrees Celsius overnight. But for long-term storage, you should adjust the pH to 7 or 8, and you want to add 1 millimolar EDTA, and you can store it in the 4 degrees Celsius or the -20. But I do not recommend storing it for a long time. Storing it overnight is okay, but if you get this far, you should keep going. Now, after this, you want to add the primer concentration that you made earlier, which is just to reiterate, it's 0.5 microliters of forward primer, 0.5 microliters of the reverse primer, 1 microliter of supernatant, 10 microliters of 2x master mix, and 8 microliters of ultra pure water. You want to add that again, and then you want to put it in the PCR. And after PCR, you want to run a gel on the PCR sample that you have. You can refer to the video on running a gel in order to do that. So just to recap everything we did in this video, explain the materials and methods needed to run the TRISOL protocol, and then explain the TRISOL protocol that we used this past year.